Q&A at the end of the all presentations. Simin Liu from ground. You'll be presenting an integration of basic molecular science and population science as a methodological advance to improving the understanding of.
So thanks a lot, Cyril. So everybody can hear me. So I'm Lyle, and it's really nice to be here. And I think 20 minutes for those of you who have attended the prior DNSG meetings would see this as a continual discussion. The way that I, really 20 minutes, and I really would like to sit in the audience and listen to all these rising stars and like Vasenti and Clement and Andrew coming after me to talk about really is pushing the most cutting edge and frontier of nutritional science. Now we have come to learn about food as medicine. Studying nutrition, just like epidemiology, studying anything in free living human, and we all recognize how difficult it is. It's not like lab animal, particularly for those of you who have feed mice and then control it and take whatever tissue you want, you know how fun that is. But for studying human, it's like a detective work. So you really have to, they might be lying to you, or you can lock them up, and then it's not ethically possible, as Vasenti mentioned. So so we have been doing this for so long now, and this is some of the reflection, as I mentioned in the past few sections. Every year, I get 20 minutes to reflect on this. The way I see this, and also as biomarker, everybody's talking about biomarkers and how we can think about all this basic molecular science can inform what's going on in human and ultimately improve not only individual but population health. So this is sort of the layout I want to talk about. And the bottom line is that one has to look at the totality, consistency, and quality of the evidence. And I've been using this slide for 20 years. I keep using it again and again. Why? Because not only do we need to be aware of all sort of biases that come almost naturally with any of human observation, be it confounding, selection, or measurement, but we also have to be mindful about nature's very true to which we have very little control, like the principles of biological interaction as well as variation that also confound us. In other words, even if you do a perfect study with perfect design, you do not expect to see the same result. And you could also be misled and lead to different conclusions if you don't appreciate these two fundamental principles of biology. So we have to be mindful in these two fundamental principles. One is interaction, another one is variation. In fact, as I will show at the end of this particular talk, is down to the molecular level. The famous particle physicist Feynman, or Caltech physicist or Nobel Prize physicist who studied particles at the quantum level, also coined the famous term about all mass is interaction. So with that in mind, obviously, it's also 2 or 3 years ago, if I remember correctly, in Athens, when I challenge or be a little bit more provocative to say even the Bradford-Hill criteria is not enough. In fact, each one of the criteria that he so famously articulated, laid out, is neither necessary nor sufficient for causal inference, perhaps with the exceptions of temporality. In other words, if you can guarantee A occur precede B, then that causal criteria is very, very necessary, but still not sufficient to demonstrate causal relationship. And as a result, for diet is such a human activities so integrated in one's life, we have to look at the coherence again across different level. Some of the level, each of those level may actually follow certain principles that are slightly different. And it only when we look at them as a whole in an integrated manner and look at its coherence, do we have some confidence in saying whether or not the things that we look at is indeed a causal relationship. Now, I just wanted to also, but the technology now, in fact, for the first time, do push us to this, the ability to look at this now. Now, as an epidemiology, obviously, nutrition is just one specialty of epidemiologists. Epidemiology, the definition is very easy to articulate, which is to study the determinant as well as the distribution of any health outcomes in human population. It's just as simple as that. And as such, we wanted to improve measurements of the exposure, mediator, modifier, or outcome. If you cannot measure, and you have to select the right population to do the study, and then you want to predict the outcome, and then you ultimately want to affect not only individual person's health outcome in a personalized manner, but also the population health outcome, and also like you said, public health. And now this stuff is just so consistent for literally over centuries now. And on my way here, and I was tempted to just put in the question of, well, can you prepare a 20-minute presentations on behalf of Professor Simin Liu at Brown University based on the stuff they've been talking about for the past 30 years onto ChatGPT. And then they actually put out some of the things that then lead me to modify some of my slides. And this is one of the things that they suggest. For those of you who are familiar with also the definitions of molecular epidemiology, they also, this is like 20 or 30 years ago, have basically identified this kind of black box approach, get into the black box, picking up each traces during, again, a period of time. And time is, again, the most fundamental parameters in defining any risk. Without this parameter, one cannot really define parameters, define risk. We keep talking about relative risk. So if you cannot define time, then you cannot define risk, let alone relative risk. And time, my friend, even physicists until today still cannot define time, right? So I just lost 6 hours times on my way here. And so right now you can tell I get hardly any kind of sleep. So maybe a little bit cognitively impaired. But that said, the technology allow us to pick up genetic, epigenetic, and then with this time that kind of give us a glimpse into this black box. And now we can, as a result, do that. And how to incorporate into this is a real challenging task, but at the same time, also extremely rewarding. And you already see the two peers speaker presenting some rewarding experience. The way that this is also the way that my research group trying to do in the past, all my career now, is the way that we see this, we need to have some large, high quality and comprehensively phenotype source population. And you heard a lot about cohorts. And I purposely exclude the nurses' health study. There's other this kind of large cohort studies out there. One is the Women's Health Study, Physicians' Health Study, also the Women's Health Initiative. Those are actually clinical trial, large clinical trial that, once upon a time, people are still, the funding agency is still willing to fund this kind of large scale clinical trial in North America, although this kind of studies are becoming rare and rare. And after the source population is defined, and this randomized control trial can also turn into subsequent observational study. So it's in a nested manner, you can even more carefully to characterize the individual involved in this population. Then you can measure biomarkers, right? Biomarker all the way from transcriptomic, metabolomic. You also heard about proteomic, and many, many more. And at the end of the day, should we stop there as epidemiologists or as nutritional science? And oftentimes, most people would do, and then go back and look at other associations of interest. But what I'm trying to encourage the sort of young investigators with the ambitions and ability to lead team science is to know you should work with folks and with the same well-defined question, continue to push the envelope so that you can really get at that, again, totality, consistency, and the quality of the evidence upon which then you can make your causal inference, which means you need to do still need to do experimental study, although may not be in as large a scale or in a different population to help verify your finding. And this is the kind of paradigm that I encourage folks to do. So I can skip these slides. You also heard about the modeling. The actual modeling is actually quite straightforward once you characterize the variable, particularly now with the computation advancements that really allow us to build tremendous amount of models with tremendous amount of parameters to really precisely characterize any population of interest. And this is actually follow a very comfortable mathematical framework, which is the so-called law of the large number, which is also been around for centuries. Basically guarantee that average of many independent random variables with mean and finite variance would be stabilized at their common means, and its convergence can be obtained through the central limit theorems. So if you look at this, you don't even need to ask folks to share data anymore, right? That's how generative AI or AI is doing. As long as you publish the paper out there, that's like why we are doing the two-sample Mendelian randomization now. It's as long as you can parameterize, as well as you correct those variables and fit into your model, you're building these density models that to articulate suitable weight. A weight is a probability that range from 0 to 1. So this is another example. And then you should also no longer need to continue to argue whether or not you should use a fixed effect model or random effect model, as some of the leading statisticians would still trying to bill you for their consulting fee, whether or not you should do a random effect model or a fixed effect model in your meta-analysis. They're all the same. If they can convert, you must well do them all. So this is the bottom line. So how do you fool around with that weight would be the major issue. The weight in terms of used to with parameterize that measure. Now, this is some of the precise measures of the diet, and this is the latest publication from Nature Food, which I find very helpful. Is that you look at the proteomic, look at the genomic, look at the metabolomics, actually at the dietary composition level to improve our measurements of dietary composition. Then I would use, and all the way integrate them to the population, in fact, at the tissue level. And these kind of things are all doable now. And this is the latest, if you kind of integrate it in the precision nutrition, the whole body level, there are increasingly a lot of the variable. You can also consider this is from as biomarker. And this is coming from my colleagues at UCLA who did this work, and ultimately capturing literally your entire biofeedback, which is interacting a human interacting with the environment in real time. And diet, as we measure based on food frequency alone, obviously is not enough. And particularly now, our understandings of the molecular sciences also allow us to appreciate even more factors affect the circadian rhythms and thus the so-called corneal nutrition here show that, in fact, different time, again, time, difficult to measure, based on the timing of your intake, the effect of which of our diet to our body might be different, and all of which ought to be captured and could indeed be captured. Now, I wanted to use a very simple, in fact, this is the most classical, in my view, almost back to the future 40 some years ago now, using the glycemic index as a way to basically illustrate the principles that I have just articulated now in the most cutting edges of doing personalized precision nutrition. Glycemic index is among the first to do this kind of work. Think of glucose, plasma glucose, capital B glucose measure as a single bioassay to capture a very complex phenomenon that go through this sort of like integrated physiology in the bodies, that you have the chemical, last night we were having these conversations over wine, chemical identity, sugar identity, whatever the carbohydrate containing foods in its either innate form with cell wall intact or in a refined form would be interacting with the matrices itself of how basically you ingest those food in your body, all which would go through our GI system. And then you can build it in the microbiota and that way probiotic and whatnot, and ultimately come up with, at the end of the day, some physiology function, which as simple as glucose here. To measure, am I using my 4 minutes to finish another 10 slides, like I said? So this is super simple. It's a black box approach, a bioassay that allow capture GI. But GI, nevertheless, is a unitness ratio measure that I've been saying this for at least 20 years. There's no need to criticize the GI because you have individual variation, because those criticism or those so-called witnesses of the GI is no different from you criticizing a consumption of sodium intake. It's also variable. It depends on individual and also depends on biological interaction. The most important of all is how you capture this kind of phenomenon, postprandial response, and ultimately integrate it with not only plasma biomarkers that can be experimentally validated. For example, feeding your mice, feeding your people under controlled environment with high carbo diet versus high fat diet. You see this kind of change of triglyceride, CRP, and also HDL, high density lipoprotein changes, which later can be in the same source population, later can be sort of like translate into greater risk prospectively of development of coronary disease. And you can look at interaction, you can look at food group one by one. All of this now it's easy to do, and you have see some interaction. In fact, you can name every single, this work is 10 years old. So I'm very proud to say in all humility at the same time that we were the first to do this kind of works, looking at pathway, looking at the whole genome assay along with the phenome assay, and trying to see whether or not we can find some of the key drivers across the metabolic pathways. And you can see across multiple diverse population, identify some of those, then later we translate back into individual dietary pattern, observationally in well-established cohort. But that's still not enough. You still have to do experimental study to validate it. In other words, now this is the magnesium work that we show. We identify different genotypes that can control the metabolism and absorptions of magnesium level that related to the effect of magnesium, and ultimately you back design in the future. In fact, I should say in the future, it's already happening now. I am sitting on a advisory board of a large trials happening in China where they first genotype folks with their and one of the enzyme called MTHFR enzyme to identify genotype, and based on their genotype, then implement intervention strategies, whether or not they should supplement with folate or not. and follow them over the course of 5 years to see whether or not their effects on not only folate level, homocysteine level, or blood pressure level, but ultimately the effects on clinical cardiometabolic endpoints such as stroke. Anyway, so let me just end with this slide. So basically, is the modern science from population science, clinical science, or basic science that come to this point. It's like the convergent point that allow us to do this. And it's really is never a better time to be a scientist that, you know, study human health and disease. And that is allow us to, nutrition, obviously take advantage of all of this. So it is very difficult to do nutritional challenge. I used to say challenging, but it is also extremely rewarding. But we do it in such a way now, we need to do it across the lifespan. In fact, should go out there and do more populations. You can do studies that across different generations, study families. Ultimately, this is my unifying theory moving forward. So we know ultimately that in the universe, there's just this many elements that define life. And we need to measure them first now. So in fact, that's the past 10 years. That's what we have been trying to do, measure all these elements in blood, in urine, in human population, and then use this information and ultimately to integrate, to enhance our understandings of all this association out there. I'm just very quickly showing this data. We do identify huge variations across population in China, in USA, in Canada, and in Brazil. And even in placenta, some of those studies need to be done. In fact, we just finished another study in Africa, in Ghana. Anyway, and this might take home. Diet is about life and food is medicine. There's no gold standard method to measure diet and night. As such, we need to do a lot of good things. And with that, I really appreciate your patience.